Good day and welcome to Scholars Universal School. To online class for SS1 Science. Today we are having biology, and under biology we'll be discussing about microorganisms in action. In the last few classes we've been talking about microorganisms. So now we're now looking at how the microorganisms act in the body. So when they are now fully transmitted from the carrier to the host or the person they carry the microorganism to. We are now looking at what will happen to the person when the microorganisms are now in action. So the growth of microorganisms like all living things do increase in size, multiply in number of cells using either the culture medium provided or any suitable surface such as moist bread as source of food. So microorganisms like other living things dey grow in the way they now grow is now divided into two. Either they have a suitable surface for where they can grow without any damage, or the culture medium provided. Like in the last class we talked about culturing and the aspect of culturing. So microorganisms can also increase in mass. Such an increase in mass or number of cells is regarded as growth, because their growth is an irreversible increase in size, mass of an organism, so they also grow. If environmental conditions of growth such as food, adequate temperature, humidity are favourable, such an increase in mass, size and number of cells of the colony is index of growth of microorganisms. So when environmental conditions are suitable for microorganisms to live in, they would they would also increase in size, mass and number of cells, so they increase in their quantity, leading to growth. So I hope you've understood the growth of microorganisms. Now let's move to the next slide. There are now how do we now measure growth in microorganisms? There are two ways. We have the first method and the second method. The first method is using the culture medium. A bacterial sample is inoculated into a nutrient agar. We all know what an agar is. An agar is also called a culture solution. As the bacterial population increases, the clear liquid medium becomes cloudy or turbid. Yes, when you introduce a bacterium into an agar, the suitable conditions of the agar will now make the bacterium to increase in number. So as they are increasing in number, the clear liquid medium will now be cloudy. You will be seeing particles inside, or turbid. The progressive turbidity now indicates a related increase in the number of bacterial cells. As I have said, the cloudier the clear liquid becomes, the more turbid it gets. So the number of bacterial cells will now lead to an increase in turbidity. In the laboratory, turbidity can be measured with a spectrophometer. This instrument measures the amount of light that can pass through a liquid medium. So as the turbidity increases, the amount of light that can pass through the liquid medium decreases. In ecology, where you guys were taught turbidity, you were taught about some ecological concepts and how they are being measured. You talked about turbidity and they said you can measure turbidity using the Secchi disc. So in laboratory now, what they use here is spectrophometer. They will measure the amount of light that can pass through a liquid medium, and as I said, as the more the turbidity increases, the less amount of light that can pass through the liquid. So if an amount of light can pass through the liquid at the beginning stage, at the mid stage the amount of light will be reducing, so leading to increase in number of bacterial cells. So the more the turbidity, the more the number of bacterial cells present in the agar. So thus, by measuring the turbidity of a bacterial culture in a nutrient agar at regular intervals, one can measure the bacterial population. So you have to now observe and record how the turbidity is increasing. So as you are calculating how it is increasing at regular intervals, you can measure the population of the bacteria present in that solution. The second method now is now a bit confusing, but with explanation you understand. In the method, small samples of bacteria taken from the nutrient agar at regular intervals of time. So as they are becoming as the bacteria are now becoming much, you now be taking small samples of them at um regular interval of time. Den dis samples are now diluted several times. Dat means you dilute dem several times, you dilute dem. Each diluted sample is inoculated onto a nutrient agar medium in a petri dish and incubated. So in a petri dish, the nutrient agar medium is now inoculated and incubated. You had already understand the meaning of incubating. Dat means they put dem in a dark place, a dark and warm place. The number of colonies formed in each petri dish is counted. So they count the number of colonies formed, as each colony is formed by multiplication of a single bacterium. Dat means it is a single bacterium that would lead to increase of multiple bacteria. So the number of colonies indicates the number of living bacterial cells in the diluted sample. So from this actual number of bacteria in the original sample can now be calculated. So after calculating the how multiplications were made from a single bacterium to the colony, we can now get the actual number of bacteria in the original sample. So you can see how the second method works. So you can use either the first or second method to learn how to measure growth in organisms. So let's now look at the beneficial effects of microorganisms, because we said some are beneficial or harmless while some are pathogenic or harmful. So some microorganisms, especially bacteria and fungi, are beneficial in three major ways: in nature, in medicine and in industries. In nature, how are they beneficial? Compost formation, nitrogen fixation, main ten ance of soil fertility, digestion of cellulose, decomposition and silage making. Nitrogen fixation in the nitrogen cycle, where there are bacteria present in helping in fixing nitrogen into the soil, and there are some that will lead to removal of nitrogen from the soil to the atmosphere. Main ten ance of soil fertility through the cycles, the microorganisms have been maintaining soil fertility. Digestion of cellulose. In this digestion of cellulose, they are present in some animals, especially the herbivores. So as they are feeding on leaves or wood, especially the herbivores, especially when they are feeding on leaves, the microorganisms help them in digesting cellulose present. And also in termites, they have microorganisms present in them which help them to digest cellulose present in wood. So they help in digestion of cellulose. Decomposition: when an animal dies or an organism dies, they help in decomposing the animal, which leads to remove discharge of nutrients into the soil. Silage making: this is making is used in agriculture in making fertilizers or making providing nutrients to the soil. While this compost formation is the after the faeces of farm animals are now packed together, and the microorganisms will help in breaking down of dis faeces into nutrients for the soil. And in medicine, they use it in manufacturing of drugs and vaccines, sources of vitamin B, sources of enzymes. So they are used in medicine for these three major ways. And in industries, they are used for baking, preparation of alcoholic drinks, making of cheese or yoghurt, writing jute, curing of tobacco, tanning of hide and seek. For baking, in baking we use yeast, and yeast is a fungi. So it helps in raising the dough and making it light. While in preparation of alcoholic drinks, we also use yeast. Yeast here is now undergoing the process known as fermentation. It helps in fermenting sugars into alcohol. Making of cheese and yoghurt, they are used. Tanning of hide and skin, that means after killing an animal, the skin, they use microorganisms in making them hard and able to be sewn for use. They use it in fashion for bags and other aspects. The harmful effect of microorganism: dey cause disease, the spoilage of food, deterioration of materials, causes of death, causes of undesired quality of product and reduction in yield. Cause of disease in that dey cause diseases, because if you look at the last class where we showed a table, where the table was showing the vector, the microorganism and the disease cause. Most diseases are caused by microorganisms. So spoilage of food dey spoil food. If there is no presence of microorganisms, food can be stored till anytime, but microorganism will now be growing on the food, thereby spoiling it. Deterioration of materials: dat means dey will cause loss in value of materials. Materials that are easily that can be easily broken down, so they will lead to loss of value. Causes of death: dey also cause death by killing living organisms, causes undesirable quality of product, reduction in yield. Dis reduction in yield is also disease. Disease of plant: like a farmer who plant yam, after pathogenic microorganisms or harmful microorganisms eat half of the yam farm, there will be reduction in yield. So that's part of their harmful effect. So now let's look at the definition of terms associated with microorganism. We have disease. Disease is a condition in which the normal function of an organism is impaired. So when an organism is impaired, means that the organism can't do some things it normally does. So this that means we say the organism have a disease. So the condition where the organism is now impaired is known as disease. Health is defined as a state of alertness, freedom from ill health, accompanied by unhindered growth and productivity of plant and animal. So when an animal or a plant is at the state of alertness, free from ill health or free from any diseases, and it is being accompanied by unhindered growth and is going at the rate where it's supposed to, as where it's meant to go, and it is giving and it is producing or being productive, it is known as it is in a state of good health. So symptom is the observable effect on an organism caused by the presence and irritation of a disease causing agent known as pathogen or disease causing agent pathogen. The symptom is now when the microorganism or the disease is now in action. What you can see in the organism that is being affected, what you can see, um what you can observe, it is known as symptom. So that's why this coronavirus, when they say that the symptoms are excess coughing, inability to breathe, we will say that the coronavirus present is already in action. So when the microorganism is now in action, what you see the person to do is known as the symptom. Pathogens also called causative agents are disease inducing microorganisms into that are now passed into plant and animal. So what or the microorganism now that is now causing the disease is known as pathogen. So when this pathogen is now reacting, the organism will show symptom, and at the end the organism will fall sick, or in some cases the organism dies. Let's look at some other factors. We have the vector here. In the last class we discussed about vector, but let's still discuss about it here. Vector is an animal which transmits a disease causing organisms, pathogen, from the victim of that disease to another person. So as I said, the vectors are just carrying the disease from one place to another, or from the affected to the non-affected. So the vector is now carrying the microorganism from where it has been in action to a place where it is not in action, so thereby causing harm to the organisms that the pathogen is passed into. Antibody is a protein molecule produced in response to a particular foreign substance or antigen. So the antibodies help in fighting microorganisms. The microorganism, when introduced into the body, the cells dey alert the brain, then the brain would lead to formation of antibodies, and these antibodies will now fight against the microorganisms, giving them resistance. So as the coronavirus has not found vaccine, most of you are wondering that how are the patients that they said they are cured, how are they cured? Is due to production of antibodies. So these antibodies will help in fighting the coronavirus, thereby leading to get the patient to be getting better. Antigen may not be a protein molecule, and it stimulates the formation of antibodies. So these microorganisms, they are known as antigens, and they lead to the formation of antibodies. So when they are produced into the body, they lead antigen lead to immune response or immunity. This immune response is now meaning that at the rate which the antibodies are now produced, so as the antigen is getting into the cell, the cells alert the brain, and formation of antibodies will take place, and the antibodies are found in the plasma fluid of the blood. So this is where they are located, and their protective function, as I've explained. So I forgot to tell you their location, but now I said it so you can understand. Vaccination is now the introduction of a mild form of pathogen into the body of an animal so as to develop resistance immunity against that pathogen as the animal grow or develop. So vaccination is now that, like that cold let me use the example of Ebola virus. You know that during the time of Ebola virus, they said Ebola virus has a vaccine. So this vaccine is not the cure. They will just inject the vaccine into people. So the antibodies will now fight this Ebola virus, but the Ebola virus is not in action. They've removed what will cause Ebola virus to react in the body. So they will remove it. They'll just introduce the non-reactive part into the body. Then these antibodies will now fight it. So the antibodies fighting it, in case Ebola virus now enters the body, the antibodies used in fighting the vaccine or the mild form of Ebola virus, the um antibodies will now kill Ebola virus. So you can see that that's why they are looking for vaccine for coronavirus. Once the vaccine has been seen, they will now be injecting people the vaccine. So as they are injecting people the vaccine, it's not that they cannot contract coronavirus, but when coronavirus is into the body, the antibodies used to kill the vaccine or the mild form of coronavirus would now be effective. They will now kill the coronavirus. So that's the meaning of vaccination, and I've defined close to 8 terms associated with microorganisms and how vaccination protects the body. Let's look at how that, because I've been talking about it. So now let's learn how it does it. The vaccine or the attenuated pathogen, that means the weak pathogen, is administered into the body. The body responds by producing copious antibodies. Then the next time the body is exposed to the same antigen or infection or pathogen antigen, that means when the real infection now enters the body or the body is exposed, the memory cells will now, because there are cells in your body that are there to store memories of how they fought a disease in the body. So the memory cells allow a more rapid and heightened or vigorous response. That means to lead to quick response in the body to antibody protection. So this antibody will now be the antibodies used to kill the vaccine, then will now be produced to fight the infection, the real infection. So it serves as a protection of the body against future infections. Some physical and chemical barriers that can prevent disease from entering the body. The physical barriers are the skin, the hairs, the cuticle, the epidermis, the bark, the cell wall lining. The chemical barriers are the mucus in the nose, the saliva in the mouth, the gum implant, tears, gastric juice or HCl, intestinal secretion. So the tears also help in preventing microorganisms from entering the body. So thanks for listening. Till we meet in the next class. like and subscribe to this channel. Contact us on WhatsApp, the number being given, and go to our website www.scholarsuniversalschool.com for more information, and go to the WhatsApp, or go to WhatsApp using ask the number for further information. Thank you. Who is that?